Great, fantastic. Pretty very nice thing. This rationally designed to big for forward, very small for backwards, and the laser pointer is here. The first speaker after me is Andy Worth. You're there. Second one is Anand is not here yet. Yes, you're there. Parak.
Not here.
Not yet. Salachi.
Yes.
And Wan Jung Ni. Not yet. Fine. I have time. I still have 7 minutes to go. Is my watch correct? Come on.
Your talk is over in 7 minutes, Giacinto. You were supposed to start at 15.
Oh, wow.
We're late.
Oh, wow. I didn't understand this. Because the session will start at 8:30. Okay, fine. Then I will start my talk. Optimizing theoretical peptides, protein interaction supported by an experimental proof that it works. Okay. This is one of the topics derived from my ERC. And there were basically 3 groups independent from me. They got from me the general directions only. And this one, the work of Sara Fortuna. Her husband, now husband, Miguel Soler. Sid Dogmo from Cameroon. Aristeo Madeira from Mexico. And Anna Russo from Napoli. I've written a lot of stuff that I do say, so if you can read it and spare me the problem of being understood when I speak. We use simulations coupled with molecular Monte Carlo optimization and molecular docking calculations. We use the result to design and eventually construct the type of molecular nanodevices. Very sensitive protein detection and synthesize a new kind of molecular drug used for instance selectively passivate enzymes. Can I be understood? Why very sensitive protein detection? There is a dramatic need for biomarker for sickness, for illness, and disease biomarkers. Protein provides often the best candidates. For instance, my own case, Parkinson, progressive supranuclear palsy, and SMAP, multiple sclerosis, Parkinsonism, are all mixed together, symptom-wise, and very difficult to distinguish. It would be nice to have a biomarker, for instance, tau protein, to measure it and decide which one. Because the first one is a synucleinopathy, the second one is a taupathy, the third one is a synucleinopathy too. So if you find alpha-synuclein in your blood, it's one thing. If you find instead tau protein, is another. We design nanobodies. We optimize their interaction with the protein of interest. After that, we use DDI, linear direct immobilization of protein and antibodies. First seed on very flat surface regions of single-stranded DNA, subsequently dock complementary DNA conjugated to antibody, we recognize the protein. Various operations are detected by the surface with a stable and sensitive atomic force microscope. Increasing the sensitivity of the last step by docking a secondary antibody conjugated to a suitable particle, we hope to achieve femtomolecular sensitivity, the so-called digital ELISA test. A new test 2 years ago came out. It's called Quinoa test. It achieved femtomolar sensitivity by means of small beads coated with antibodies. They see the protein, they dock on top of it a second antibody, and then they make it fluorescent. Only those beads which are one protein on it, there are never more because the protein is very diluted, can become fluorescent. When you put in a filter, separating them all of them in one little hole, and you have a digital screen, you can count the fluorescent spots quantitatively, and it's very nice. Apparently, it's commercially available. Among all the problems, my eyes closed at the time, and I have to open them up to talk to you. Let us start by showing the optimization if possible. Peptide-protein combination, then show that it is possible to design peptide-protein for back interactions. Because an enzyme typically is made by a cavity where the passivators go in, and a back where it's very difficult to drug anything. By designing the antibody for the back, we can achieve binding also the back of the protein. Therefore, by linking the back and the one in the cavity, we can have a very sensitive, very specific drug for that particular enzyme. These are a fortuna, effort. Antibodies, everyone knows what they are. There's a big difference in size. An antibody is typically 1,000 amino acids. Peptide is 13 amino acids, 2 order of magnitude less. So it can be designed much better. Combination limited number of building blocks, highly versatile possible combination scale 20 to the n, possibility of automated synthesis. These are the maltose binding protein. Maltose binding protein because I had used atomic force microscopy in the past to use maltose binding protein and show that it was binding sugars and things like this. And we published 2 papers. CLINAM was excellent. Binding papers for very weak binding. We had excellent CLINAM, so I decided to try atomic force microscopy also for that technique. Francesco, when I'm 2 minutes past 10, you tell me, please. We measure binding site, no binding site, the binding energy of various peptides, choose the best one binding. And binding affinity determination was done with SPR. This binding affinity is 416 plus minus 72 micromolar, not big result. Docking it was done not with simulation, it's using only docking programs. Docking programs are notorious for being imprecise. But in order to be able to use molecular simulations, many seconds of it in the simulations, many nanoseconds, we move over to a smaller protein. It is possible to design for a sulfur binding site for the outside of the protein. If you don't include a solvent in the sulfur binding site, you don't get any bonding because most of it is hydrophobic bonding. But if you do include the solvent, both atomically or an effective solvent, you can do it. And this is what we did, peptide generation without solvent, computational screening, molecular solvent, experimental screening, binding affinity determination, binding identification, application protein mobilization. Okay, here is the docking method as usual. You go down, you go to a huge binding energy is too good. The docking problem, I told you before, there's a big discrepancy between the theory and the experiment here, but however, they go down both. So if you are showing binding for the docking, there's binding also for the experiment. Two peptide bonds will be, this one is in the cleavage of the molecule, and this one is instead, no, sorry. This one is for the back of the protein. To do experimental proof, we graft in a small micron patch of peptide. Next slide will be more clear. You see what we do. Graft in DNA. Then we use DNA conjugated peptides. Put them on a flat surface, very flat surface, and target beta-2 microglobulin comes and shows some increase in height. Using this method, we also, for prime protein, show the different orientation for two different antibodies. The molecule docks like this, the molecule docks vertical, and it was easy feasible to see the results with atomic force microscopy. You see, the synaptonoids don't look very good, not very flat the surface, but still, if you notice that there is correspondence to each height, down, up. If you go through it, the two errors become quite small even. The surface is not very, very flat. And with some care, I can guarantee you, the surface can be made flat. Therefore, the synaptonoid can be improved. Here's the binding. Again, for beta-2 microglobulin, the binding was poor, 100 and something micromolar, but a good antibody binding to a protein will be nanomolar typically. In this case, it's not. We didn't succeed in going further. And then you see here, here's the size of the protein. If it would dock vertically, we see 4 nanometers, but since on the side, 2.5 nanometers. And here, again, the surface roughness, if you follow the surface roughness, see that the synaptonoid is much better than what it looks in this slide. Okay, this is, then we decided to simulate with the beads a binder to find out what's the, finished now. We come to the final slide where the rigid, flexible protein to rigid binders, to rigid hooks are better than a rigid, this is better actually, a rigid binder, sorry. Computational design corrected target chosen binding site, protein being at the pocket easily, where surface protein on druggable site difficult. This is an advantage feature that can be exploited for oriented protein immobilization on surfaces. Ex novo immobilization of peptides, starting from a polyalanine, can reach microaffinity also for surface protein binding site. This is important because 2 microaffinity combined can put you into the nanobody, into the nanomolar range, and maybe further. Stay tuned for the developments. Thank you. I apologize. It's the first talk I give in a long time, and my way to speak is very poor. I realize it. Very soft, but that's what it is. Questions? Questions? No question? Move on to the next speaker. with Underworth, computational toxicology, lesson learned from the now compute project.